Hello everybody and welcome back to my YouTube channel. And अगर मैं आपको बात करूँ आज के इस topic की, so today's special topic is rapid revision and a quick recap for pharmaceutical biotechnology. So pharmaceutical biotechnology has multiple domains from which question can appear. One of them is fermentation products. So there are a lot of different microbes or that are used for fermentation, like Lactobacillus leishmania for vitamin B12, and there are different other Acetobacter aceti for vinegar and all. So you need to know different fermentation products as well as recombinant DNA products like humulin, insulin, right? Then erythropoietin, hemopoietin, all those things. So it is very essential for us to know about the different products that are used in gene therapy as well, if you can remember any products. rDNA technology. In rDNA technology, you need to know about what are vectors, plasmids, cosmids, bacteriophage, yeast artificial chromosome, bacterial artificial chromosome. Then you need to know about the DNA ligase ka role, right? Because it's very essential to know about the DNA. Then you need to know about the molecular scissors, which are known as restriction endonuclease. What are sticky ends, staggered ends, blunt ends, sharp ends, and all. So that is very essential for us to know that we are well versed with all those things. So make sure that you know about these topics अच्छे से. Then when I talk about the central dogma, so in central dogma, transcription, translation, replication, there are different enzymes that are involved. Reverse transcriptase is involved from conversion of RNA to DNA. So all those things are very essential. And Okazaki fragments, role of RNA primase, types of polymerase. You need to know different polymerase, the types of RNAs, and the two models for tRNA, cloverleaf model, and all. So it's very essential to know that. Then biotechnological techniques. These techniques include ELISA, RT-PCR, only PCR, blotting, immunoblotting, which is southern blotting for DNA, western blotting for protein, and northern blotting for RNA. Then different types of siRNA, miRNA, microRNA, silencing RNA, small interfering RNA. So what is a PCR? Different steps of PCR: denaturation, renaturation, polymerization, the different temperatures, thermosaquaticus polymerase is used, and then microbial genetics. So from microbial genetics, mostly the questions of your from transformation, transfection, transduction, conjugation. So these different processes like there's microinjection, biolistics and all. Similarly for transformation, bacterial transduction is for viruses and transfection is in eukaryotes. When we talk about transposons, so there are two types of transposons, DNA and RNA transposons, which can be further divided as replicative and non-replicative. So replicative are basically copy paste, while non-replicative are cut paste. So you need to know about the different types of then palindromic sequences, right? All those things you need to know about that. Then bacterial growth cycle. So what is the different phases? What is the role of different phases? Initiative, lag, log, exponential, stationary, and then death phase. Then the last and the most important is vaccines and immunology. So questions may arise from hypersensitivity reactions, type A, 1, 2, 3, 4, anaphylactic, atopic, retarded, cytotoxic, which is cell mediated, which is immune mediated, which immunoglobulins, when the structure of an antibody, different immunoglobulins. So in that monoclonal antibodies ka production is very important, right? Hybridoma technology, PEG medium, HAT medium, PEG is a fusing agent, myeloma and B cells combine, and all those things. So it is very essential for you to know. Immunoglobulins IgG, which is the largest, which is the most abundant, which is the first in primary response. Which one is the dimer and monomer in serum and milk? Which one is responsible for allergic responses? So all those things needs to be known. Vaccines, may if I tell, we have live, attenuated, inactivated, killed, different kinds of vaccines that are available, subunit conjugates, DNA vaccine, RNA vaccine. You must know the examples of different vaccines. Polio, measles, mumps, rubella, which is bacterial, which is viral, like oral polio vaccine, Sabin is live, like while parenteral polio vaccine, IPV is Salk, which is not, which is killed, right? So if you need to know the different vaccines, then toxoids, diphtheria, right? Tetanus toxoid and all. You need to know about horse serum, the role of horse serum. So all these things, the biotechnology में questions arise: hybridoma, monoclonal vaccines, immunology, and then MHC complexes. The difference between MHC1 and MHC2, major histocompatibility complex, which is CD4 cells. So MHC2 is related to CD4, and it is a helper, while CD8 and it is killer. So which one is antigen presenting cells? So dendrites, macrophages, B cells are antigen presenting cells. You need to know about them as well. It's very essential to know about the APCs. So that is a very important point of view. Then obviously I mentioned about the different techniques, ELISA, Western blotting में what is blocking agents, what is primary antibody, what is HRP substrate, TMB substrate, what is the color change, what is Ponceau staining, just a glimpse, that's it. What is the nature of the blot membrane on which it is transferred, which is nitrocellulose or PVDF, polyvinyl difluoride and all. Then RDNA technology may you must know about the recombinant vectors available, different sizes of the vectors, advantages of cloning vectors, right? So cloning is very essential. So that is something that you need to know. And fermentation products, as I already mentioned. So that's it from biotechnology point of view. I hope this video was very insightful for you and you would have got to learn a lot from the video. Do like, share, subscribe my channel for more such upcoming vlogs.